Morning everyone. Today we are going to start microbiology portion. Phylogenetic overview and general features of prokaryotic organisms. The basic physical unit of living organisms is a cell. Biologists organize and recognize two types of cells: eukaryotic and prokaryotic cells. Eukaryotic cells contain membrane-bound compartments including a nucleus and organelles such as mitochondria. Chloroplasts, prokaryotic cells, lack a true membrane-bound nucleus and extensive internal compartments. Prokaryotes are distinguished from eukaryotes by the absence of a unit membrane-bound nucleus and usually the lack of their other cell organelles. Three-domain system is there. About the classification, historically, prokaryotes were classified on the basis of their phenotypic characteristics. Prokaryotic taxonomy therefore involved measuring a large number of characteristics including morphology and biochemical characters, ability to grow on different substrates, cell wall structure, antibiotic sensitivities, and many others. This contrasts with classification of eukaryotic organisms, for which phylogenetic evolution-based classification was possible through the availability of fossil evidence. A major evolution occurred with the realization that evolutionary relationships could be deduced on the basis of difference in gene sequence. The most important gene for prokaryotic phylogeny is the 16S ribosomal RNA, rRNA gene, which is present in all cells. The gene is approximately 1500 bases in length and possesses regions in which sequences are conserved, facilitating sequence alignment, and variable and hypervariable regions which enable different organisms to be discriminated from one another. Based on ribosomal RNA data, Carl Weiss proposed a radical reorganization of the 5 kingdoms into 3 domains. In his classification system, Weiss, I am not sure about the pronunciation, placed all 4 eukaryotic kingdoms, Protista, fungi, plantae, animalia, into a single domain called eukarya, also known as eukaryotes. He then split the former kingdom of Monera into eubacteria, bacteria, and archaebacteria domains. Unlike Whittaker's 5 kingdom system, Weiss' 3 domain system organizes biodiversity by evolutionary relationships. So here we could see a diagram which represents the phylogenetic tree of living things based on RNA data proposed by Carl Weiss, showing that separation of bacteria, archaea, and eukaryotes from a common ancestor. Phylogenetic tree of life from which different roots are and branches are shown. This is a rooted one. No, not rooted, I guess. Aquifex, Thermotoga, Cytophaga, Planktomyces, Cyanobacteria, Proteobacteria, Spirochetes, Gram-positive, green filamentous bacteria. All these comes under bacteria section. Then archaea section is not a proper word for this, but I am using for a common understanding, so don't get me wrong. Then archaea, Pyrodicticum, Thermoproteus, T. celer, Methanococcus, Methanobacterium, Methanosarcina, halophils. And Eukaryota, Entamoeba, slime molds. Animals, fungi, plants, chelates, flagellates, trichomonads, microsporidia, diplomonads, etc. All this comes under Eukaryota. General features of prokaryotic organisms: 2 membrane-bound nucleus absent, DNA complex with histone absent, number of chromosome 1, few contain more than 1, mitosis and meiosis absent, genetic recombination partial, unidirectional transfer of DNA. Then sterol in plasma membrane absent but present in mycoplasma. Ribosome 70S. Unit membrane bound organelles absent. Cell wall made up of peptidoglycan in new bacteria absent in mycoplasma. Once again, I'm going to read these general features of prokaryotic organisms. True membrane bound nucleus absent. DNA complex with histone absent. Number of chromosomes 1. Few contain more than 1. Then mitosis and meiosis absent. Genetic recombination is partial. Sterol in plasma membrane absent but present in mycoplasma. Ribosome 70S. Unit membrane bound organelles absent. Cell wall made up of peptidoglycan in new bacteria absent in mycoplasma. So prokaryotic cells show similarities with eukaryotic organelles like mitochondria and chloroplast. The endosymbiotic theory, Margulis in 1993, proposes that mitochondria and chloroplasts of eukaryotic cells originated as symbiotic prokaryotic cells. The presence of bacterial circular covalently closed DNA and 70S ribosome in mitochondria supports this theory. The presence of bacterial circular covalently closed DNA and 70S ribosomes in mitochondria supports this theory. Comparison of prokaryotic cell and eukaryotic cell. Here we could see nature of DNA. In case of prokaryotic cell, they is circular, but in case of eukaryotic organelles, they is circular. Histone protein absent in case of prokaryotic cells, absent in case of eukaryotic cells, and there was some mistake, I guess. Yeah, so don't bother. This thing is problematic. I won't be covering this portion. Sorry. Then bacteria cell structure we are going to learn. Bacteria cell structure. Bacteria are microscopic, relatively simple prokaryotic organisms whose cells lack a nucleus or nuclear membrane. Bacteria vary in size from less than 0.2 μm in diameter to more than 50 μm in diameter. The largest known bacterium in terms of total cell volume is Theomargarita namibiensis, whereas smallest known example is Mycoplasma pneumoniae. So, largest known bacterium in terms of total cell volume is Theomargarita namibiensis, and smallest known example is Mycoplasma pneumoniae. The bacteria may appear as root, bacilli, sphere, cocci, and spiral, cocci. Yeah, spiral, spirilla, or spirochetes shaped. Bacteria reproduce by binary fission, have unique cell walls, and exist in most environments on Earth. They live at temperatures ranging from 0 degrees Celsius to over 100 degrees Celsius in conditions that are oxygen rich or oxygen free. Cell structure, cell wall. The cell wall of both gram-positive and gram-negative bacteria is chemically peptidoglycan murine, which confirm the characteristic cell shape. Peptidoglycans are unique to prokaryotic organisms. In gram-positive bacteria, peptidoglycan cell wall consists of a single 20 to 80 nm thick homogeneous layer lying outside the plasma membrane. In contrast, the gram-negative cell wall consists of 2 to 7 nm thick peptidoglycan layer covered by a 7 to 8 nm thick outer membrane. The space that is present between the plasma membrane and the outer membrane in gram-negative bacteria is called as the periplasmic space. The substance that occupies the periplasmic space is called periplasm. Periplasmic space also presents in some gram-positive bacteria. However, the nature of the periplasmic space and periplasm differs in gram-positive and gram-negative bacteria. Peptidoglycan is a polymer Peptidoglycan is a polymer which contains two sugar derivatives, N-acetylglucosamine and N-acetylmuramic acid, NAG and NAM. N-acetylmuramic acid joined through beta-1,4 glycosidic bond. A peptide chain of 4 alternative D and L amino acids called tetrapeptide. L amino acids called tetrapeptide is connected to the carboxyl group of the NAM. Amino acids present in tetrapeptide include L-alanine, D-alanine, D-glutamic acid and either lysine or diaminopylic acid DAP. Often the carboxyl group of the terminal D-alanine is connected directly to the amino group of diaminopylic acid. In some peptidoglycans, a short peptide chain called peptide interbridge connects the tetrapeptide chains. The reaction that forms the peptide cross-linking during peptidoglycan synthesis is called transpeptidation. Reaction that forms the peptide cross-links between peptidoglycan synthesis is called transpeptidation. Gram-positive cell wall, peptidoglycan plasma membrane. So here we could see structures. Gram-positive cell wall, which shows peptidoglycan layer and plasma membrane. Then in case of gram-negative cell wall, outer membrane is there, peptidoglycan layer is there, plasma membrane is there, and in between this outer membrane and peptidoglycan layer, there is periplasmic space. Outer membrane, plasma membrane, in between that the peptidoglycan layer is also there. That is the case with gram-negative cell wall. Periplasmic space is there. Okay, and the chemical structure of this NAM and NAG. Direct cross-linking peptide interbridge. Two groups of bacteria devoid of cell wall peptidoglycans are the Mycoplasma species. So two groups of bacteria devoid of cell wall peptidoglycans are the Mycoplasma species, which possess a surface membrane structure, and the L forms that arise from either gram-positive or gram-negative bacterial cells that have lost their ability to produce a peptidoglycan structure. Many gram-positive bacteria have acidic substances called teichoic acids. Teichoic acids are polyol phosphate polymers. So, teichoic acid is there. Teichoic acids are polyol phosphate polymers bearing a strong negative charge. They are covalently linked to the peptidoglycan. They are strongly antigenic but are generally absent in gram-negative bacteria. What is gram staining? We mentioned many times that gram-positive bacteria and gram-negative bacteria is there. So what is exactly this gram staining, which determines which bacteria is gram-positive and which one is gram-negative? So let's learn about that. Gram staining. Staining is a biochemical technique of adding a class-specific DNA, proteins, lipids, carbohydrates, dye to a substrate to qualify or quantify the presence of a specific compound. So staining is a biochemical technique of adding a class-specific dye to a substrate to qualify or quantify the presence of a specific compound. Gram staining or Gram's method is an empirical method of differentiating bacterial species into two large groups based on the physical properties of their cell walls. The method is named after the inventor, the Danish scientist Hans Christian Gram in 1853 to 1938, who developed the technique in 1884. The gram staining procedure involves four basic steps. The bacteria are first stained with the basic dye crystal violet. Both gram-positive and gram-negative bacteria become directly stained and appear purple-blue after the step. So first stain is crystal violet. Crystal violet is first stain. The bacteria are then treated with gram-iodine solution. First we treated bacteria with crystal violet stain and they both got turned purple-blue after this step. And the bacteria are then treated with gram-iodine solution. This allows the stain to be retained better by forming an insoluble crystal violet iodine complex. Both gram-positive and gram-negative bacteria remain purple-blue after this step. Grams decolorizer, a mixture of ethyl alcohol and acetone, is then added. This is the differential step. Gram-positive bacteria retain the crystal violet iodine complex, while gram-negative is decolorized. So Gram's iodine solution is applied along with that. Now after that, Gram's decolorizer, a mixture of ethyl alcohol and acetone, is added, which removes the color in case of gram-negative bacteria. So finally, the counterstain safranin also, which is a basic dye, is applied. Since the gram-positive bacteria are already stained purple, they are not affected by the counterstain. Gram-negative bacteria that are now colorless become directly stained by the safranin. Thus, gram-positive appear purple-blue and gram-negative appears to be having a color of red or pink color, which is produced because of the safranin stain, which is acting as a counter stain. So first, crystal violet is applied, then gram iodine solution. So first, when we apply this crystal violet, we get a purple blue color. Later, gram iodine solution is applied, which gives a violet iodine complex. But the color remains purple blue. Okay. Then Gram's decolorizer is applied, which removes the color in case of gram-negative bacteria, which is again colored with the help of safranin. And then acid-fast staining. The acid-fast stain is a differential stain used to identify acid-fast organisms, such as members of the genus Mycobacterium. Acid-fast organisms are characterized by wax-like, nearly impermeable cell walls. They contain large amount of mycolic acid. So acid-fast organisms are characterized by wax-like, nearly impermeable cell walls. They contain large amount of mycolic acid. The primary stain used in acid-fast staining, carbol fusion reagent we mentioned. The primary stain used in acid-fast staining, carbol fusion, is lipid soluble and contains phenol, which helps the stain penetrate the cell wall. This is further assisted by the action of heat. The smear is then rinsed with a very strong decolorizer, which stripes the stain from all known acid-fast cells but does not permeate the cell wall of acid-fast organisms. The decolorized non-acid-fast cells then take up the counter stain. Capsules. Some bacteria form a thick non-molecular weight viscous polysaccharide material on their outer surface. The terms capsule and slime layer are frequently used to describe these layers. The composition of these layers varies in different organisms but may be thick or thin, rigid or flexible depending on their chemical nature. The rigid layers are organized in a tight matrix called capsule. If the layer is more easily deformed and washed off, then called slime layer. Most capsules consist of polysaccharides. However, the capsule of some Bacillus anthracis consist of polypeptides. mainly polyglutamic acid. The possession of capsules of these bacteria make some pathogenic bacteria resistant to phagocytosis and enhances their virulence. And other membranes, in addition to peptidoglycan cell wall, gram-negative bacteria contain an additional membrane, the outer membrane. This layer is not constructed solely of phospholipid and protein, as is the plasma membrane. It also contains polysaccharide. The lipid and polysaccharide are linked in the outer membrane to form a lipopolysaccharide complex. Because of this, the outer membrane is often called the lipopolysaccharide layer, LPS. LPS consists of three components: the core polysaccharide, the O polysaccharide, and lipid portion, referred to as lipid A. Lipid A is not a glycerol lipid, but instead the fatty acids are connected by amine ester linkage to a disaccharide composed of N-acetyl glucosamine phosphate. An O-specific polysaccharide, core polysaccharide lipid A structure of LPS of gram-negative bacteria. Outer membrane of gram-negative bacteria is relatively more permeable as compared to plasma membrane. This is because proteins called porins are absent in the outer membrane that function as channels for the entrance and exit of hydrophilic low molecular weight substances. Outer membrane of gram-negative bacteria is relatively more permeable as compared to plasma membrane. This is because proteins called porins are present in the outer membrane that function as channels for the entrance and exit of hydrophilic low molecular weight substances. Then plasma membrane. The bacterial plasma membrane is a unique membrane composed primarily of protein and phospholipid. One major difference in chemical composition of membranes between eukaryotic and prokaryotic cells is that eukaryotes have sterols in their membranes. Sterols are absent from the membranes of all bacteria except Mycoplasma. Molecules similar to sterols called hopenoids are present in the membranes of many bacteria and are likely to play a role similar to that of sterol in eukaryotic cells. Plasma membrane perform many functions including transport, biosynthesis and energy transduction and invagination or enfolding of the plasma membrane called mesosome in bacteria that may be involved in chromosome replication or cross-wall formation in dividing bacteria. Then cytoplasm. The bacterial cytoplasm is densely packed with 70s ribosome. Bacterial cytoplasm also contains many organic and inorganic granules that may be non-unit membrane bound or membraneless. These granules may be glycogen, poly-beta hydroxybutyrate or PHB, cyanophysin granules composed of large polypeptides containing approximately equal amounts of amino acids, arginine and aspartic acid present in cyanobacteria, and carboxysome, polyhedral inclusion bodies that contain CO2 fixation enzymes, ribulose 1,5-biphosphate carboxylase found in cyanobacteria, nitrifying bacteria and theobacilli, and metachromatic granules of polyphosphate in the cytoplasm of some bacteria that show differences in color when stained with a blue basic dye act as reserves of phosphate. Then surface appendages. This is very important. Two types of surface appendages can be recognized in certain bacterial species: the flagella, which are organs of locomotion, and pili. I'm not sure about the pronunciation. Latin hairs meant for conjugation. Flagella occur in both gram-positive and gram-negative bacteria. Structurally, bacterial flagella are long, filamentous surface appendages. A flagellum consists of three parts: a long filament, the long filament which lies external to the cell surface, the hook structure at the end of the filament, and a basal body to which the hook is anchored and which imparts motion to the flagellum. The protein subunits of a flagellum are assembled to form a cylindrical structure with a hollow core. So chemically, flagella are filamentous, constructed of a class of protein called flagellins. The hook and basal body structures consist of numerous proteins. The basal body transfers the outer wall and membrane structure. It consists of a root and one or two pairs of discs, gram-positive, two rings in basal body. Wait a minute, I got a bit confused. So I'm going to read this again. The protein subunits of flagellum are assembled to form a cylindrical structure and a hollow core. Chemically, flagella filament is constructed of a class of protein called flagellins. The hook and basal body structures consist of numerous proteins. The basal body transfers the outer wall and membrane structure. It consists of a root and one or two pairs of discs, gram-positive 2 rings in basal body and gram-negative 4 rings in basal body. The thrust that propels the bacterial cell is provided by counterclockwise rotation of the basal body, which causes the helically twisted filament to wall. The movement of the basal body is driven by a proton motive force rather than by ATP directly. The number and distribution of flagella on the bacterial surface are characteristic for a given species and hence are useful in identifying and classifying bacteria. Then monotrichs have one flagellum, amphitrichs one flagellum attached at each end, then lophotrichs cluster of flagella at one end or both end. Then peritrichs. Flagella are spread fairly evenly over the whole surface. Then atrichs without flagella. Mono, amphi, lopho, peritrichs, atrichs. For example, cholera has a single flagellum at one pole of the cell. It is monotrichous, whereas Proteus vulgaris and E. coli have many flagella distributed over the entire cell surface. They are peritrichous. So pili occur almost exclusively on gram-negative bacteria and are found on only a few gram-positive organisms like Corynebacterium renale. The terms pili and fimbria are usually used interchangeably to describe the thin hair-like appendages on the surface of many gram-negative bacteria. Proteins that form pili are referred to as pilins. Pili are more rigid in appearance than flagella. In addition to the role of the pili in conjugation, they also function as receptors for donor-specific, male-specific phages. Then endospores. Bacillus and Clostridium species can produce endospores, heat-resistant, dehydrated resting cells that are formed intracellularly and contain a genome and all essential metabolic machinery. The endospore is encased in a complex protective spore coat. Diplocholinic acids. No, I'm sorry. Dipicolinic acids. Yeah, just closely look at this. That acid, dipicolinic acid, present in great quantities in the bacterial endospore. It is thought to contribute to the endospore's heat resistance. So that is very important one. Dipicolinic acid. Dipicolinic acid. Maybe the pronunciation will be different and I'm not sure about that. Sorry. Endospore morphology and location vary with species. And staining is used to determine the morphology and location. Staining of endospore through Schaffer-Fulton procedure first involves heating of bacteria with malachite green that can penetrate endospores. It gives the endospore green color. After malachite treatment, cell is washed with water and is counterstained with safranin. Cell wall, vegetative cell, plasma membrane, DNA. Cell wall is there, plasma membrane, DNA, vegetative cell, starvation conditions, axial filament of DNA, septum forms, pre-spore, spore mother cell, then space for spore coat, engulfment of pre-spore, spore coat material, then spore becomes surrounded, spore coat material deposited, mother cell lysis, spore released. Stages in development of a Bacillus subtilis spore. Okay, so closely look at this and learn. When a growing bacterium, the vegetative cell, is faced with starvation, the process of sporulation begins. The first morphological change is the transformation of the two identical chromosomes, the parent chromosome and the newly replicated daughter chromosome, into an axial filament of DNA. A septum then divides the cell, and the smaller pre-spore cell is engulfed by the spore mother cell. In the cell within a cell, the plasma membrane separates the cytoplasm of one cell from the cytoplasm of the other cell and leave a space in between. In this space, the spore coat material is deposited. Okay. So here we could see a characteristic difference between gram-positive and gram-negative bacteria. So in case of gram-negative bacteria, cell wall is 2 to 7 nm thick. In case of gram-positive bacteria, 20 to 80 nm thick. No teichoic acid present in case of gram-positive bacteria. Cell shape: spheres, ovals, straight or curved roots, helices or filaments. Some have sheaths or capsules. Spheres, rod or filaments may show true branching. Then reproduction, binary fission, sometimes but in case of gram-positive bacteria, binary fission. Metabolism, phototrophic, chemolithoautotrophic or chemo-organoheterotrophic. Then in case of gram-positive bacteria, usually chemo-organoheterotrophic. Motility, motile or non-motile. Flagellation can be varied type: polar, low food trichis, peritrichis. Most often non-motile, have peritrichis flagellation when motile. Then appendages can produce several types of appendages like pili, fimbriae, and usually lack appendages in case of this gram-positive bacteria. Then endospores cannot form endospores. Some groups can form endospores.